planet that our species lives on is Morenu. Morenu is a dark rogue planet, meaning that it is not a part of a solar system or orbiting a gas giant. The planet's terrain is similar to that of Earth, including lakes, mountains, vegetation, and few deserts. Due to the lack of light, the planet is quite cold, having an average temperature of -23 degrees, with most of the heat being generated from the friction of tectonic plates sliding over a constantly turning mantle and core. The planet experiences frequent hail and snow, and most of the animals on the planet have developed mechanisms for dealing with the cold, such as extra fat or more cholesterol within their cells. The species that our team has encountered most is Siyu. Siyu move in a swarm of close to 1,000 individuals and are free roaming. They are territorial, with swarms controlling areas of roughly 10 kilometers squared and are constantly in flight. Due to the swarm's constant movement, members of the swarm sleep in alternating cycles, with sleeping individuals being carried by others. They do this by attaching their feet and cerci, being carried upside down for an average of 3 hours at a time. While swarms have been observed across multiple biomes, they are concentrated in heavily forested areas. Siyu emit ultraviolet and infrared light for their own use, but the planet's flora utilize some of this light for growth and development. Siyu emit UV and infrared light intermittently, meaning the environment around the swarms have a constant source of light. The flora absorb infrared light and use it for photosynthesis via chlorophyll F, with the UV light both stimulating seed germination and hardening the flora's defense system, aiding defense against herbivory. Siyu are a carnivorous swarm that feed primarily on carrion, but will kill other small carnivores and medium-sized herbivores for food if necessary. Due to the animal's abundance, carrion is often consumed shortly after death, with very little remains left for decomposition, making Siyu the natural cleaners of the land.
Siyu is a small size of arthropod that exists as part of a swarm. It has exoskeleton that functions as the hard shell on the outside of its body that can flexibly join with underlying muscles for exoskeleton's movement. Now I'm going to explain the structures of Siyu. It has a relatively cute body combination. Similar to arthropods, Siyu's body is divided by 3 segments, including head, thorax, and abdomen. Siyu has no eyes, antennae, and small head to control its whole body, and it links to thorax part with 6 forefeet for stabilize the body when resting in slippery areas. For the abdomen part, Siyu has an approximately 5 times bigger abdomen and a part of transparent wings, which is similar to dragonflies. Also, Siyu has a pair of long tails located in the end of abdomen's left and right side separately. Now I'm going to talk about the physiological system, respiratory system, and digestive system. For Setia's respiratory system, air comes through a small trachea directly to the body cells. Each body cell is close to the tracheals to limit the distance of diffusion. For its digestive system, Setia's mouthpart is sharp and strong to capture and intake food. Firstly, piercing the food to smaller pieces. This action takes place in mandibles, and two pairs of maxillae are used to swallow food to pharynx. After that, digestive systems start digesting food, from foregut, midgut, to hindgut. Now I'm going to talk about the environmental interaction. Due to water near its dark world, pupil doesn't function as much as in the other world, where I have sunlight. Therefore, pupil doesn't function as much and becomes degenerated. But Setia needs to detect predators and prey for food. So what substitutes pupil? Photoreceptors. Photoreceptor cells are separated by emitter and receptors. The emitters of photoreceptor cells are evenly embedded in the abdomen region. This is because Setia have a giant abdomen for fat storage in order to resist the cold temperature in that world. While the receptors of photoreceptor cells located in the tail, the Setia is able to receive signals or detect the distance of predators when it is flying. Now, Scott's gonna talk a bit more about the in-depth descriptions of Setia's physiological system.
The light created by the Setae is very similar to the light created by the fireflies found on planet Earth. To begin the process, the head Setae will move into an upright position in the air, allowing its enlarged underside to be exposed to the elements. A quarter of the Setae will then proceed to follow the head Setae. From here, they will proceed to begin the process involved in creating a flash. The light-emitting organ found upon the gut of the Setae is transparent and, like the rest of the Setae, filled with small pores known as spiracles. These spiracles allow oxygen to attach to the surface of the light-emitting organ, where they then will bind to the mitochondria until such a time as nitric oxide can be released from neurons in the photocyte cells within the organ. Here, the oxygen will remain bound and consumed until the Setae releases nitric oxide, at which time the nitric oxide will instead bind with the mitochondria and the oxygen will be free to enter the light organ. Within the light organ, oxygen will now combine with calcium, adenosine triphosphate, and the substrate luciferin. The bioluminescent enzyme luciferase, released as the Setae determines its time to create a flash, will then bind with the luciferin and begin the oxidizing process along with the magnesium. The end result of this mixture is adenosine monophosphate, carbon dioxide, the oxidized version of luciferin, known as oxyluciferin, protein phosphatase 1, and a cold light. This cold light is ultraviolet light, barely perceivable by the human eye, ranging from 200 to 400 nanometers. This light would, in our star system, be most commonly thought of as being the harmful light that damages our skin and eyes, produced by the yellow dwarf star, our Earth orbits. The protein phosphatase 1 that is produced will bind with the adenosine triphosphate, inhibiting the substance and preventing any further light-producing reactions. The light will then proceed to move throughout the environment within a 45-degree cone in front of the Ceres swarm, bouncing and reflecting off the environment around the swarm. This light, reflected by the trees and the rocks around the Ceres, is then taken up by the photoreceptors located in the tail of the Ceres as tiny pinholes, having no discernible protective coverings on the top of them. As the photons from the ultraviolet light emitted from the Ceres bounce back into these small pinholes, they are absorbed by photoreceptor protein known on Earth as OPN5. As OPN5 absorbs the photons, a change is triggered with the makeup of OPN5, allowing it to bind with ion channels on the surface of the photoreceptor cell. This results in a hyperpolarization of the photoreceptor cell, and as a result, a change to the membrane potential of the rod-like cell. This change in membrane potential allows sodium to enter the cell, exciting the cell and prompting it to send out a pulse to the optic nerve. The optic nerve then travels to the Ceres brain, where the optic nerves reach the optic chiasm, an X-shaped structure found in the brain of the Ceres. The optic chiasm appears to be wasted upon the Ceres, however. As the Ceres has only a line of photoreceptor cells running down its tail, it does not need images superimposed in the same sense that we would. This discovery indicates that the planet Murinu may have once circled a star of its own in the not-so-distant past. From the optic chiasm, the pulses set in motion by the photons running along the optic nerves reach the lateral geniculate nucleus, where the objects around Ceres are given a range and object movement is predicted. From the lateral geniculate nucleus, the pulse is sent into the visual cortex, where the image received through the proton waves are translated and the shape, depth, and velocity are all found. The movement of both the insect and the landscape around the insect are also analysed and calculated, the second part being compared with the mid-motion cortexes. The pulse is then sent into the hippocampus, where the image is stored, although not for a great deal of time. From here, the setae is able to determine a rough estimation of the surrounding foliage, topography, as well as identify anything that may be dead or dying on the ground. By limiting the flashes to only a few seconds, the setae reduces the chances of any of its few sight-gifted predators observing the light and preying upon the setae swarm itself. Once nourishment of any kind, whether it be living or not, is located, the setae swarm upon their prey akin to that of piranhas on Earth. They are far from picky eaters. Little is left of the setae's meal after the feast is complete, with generally nothing but bones remaining. Moonwani does, at certain times of the year, have heavy rainfalls. When this occurs, the light Circe emits to find prey becomes ineffectual, and so another means must be used to hunt for prey. Circe deal with this problem with the use of a form of heat vision, not dissimilar to that used by serpents on planet Earth. Nerve cells in the pit organ upon the setae's tail contain temperature-sensitive ion channels known as TRPA1 and TRPV1, thermal noise receptors that detects the infrared heat emitting from the bodies of living or recently killed warm-blooded animals. Both the TRPA1 and TRPV1 channels are composed of 6 membrane-spanning helices with intracellular nitrogen and carbon termini with G-protein receptors included. As the organ is hit with the infrared light in the form of heat, the G-protein receptors begin the process of elevating the signal by hyperpolarizing the cell, resulting in a change to the membrane potential of the nerve cell.